So Jonathan, we're not going to like post this whole thing or anything, but at the end, I will ask you to say something nice about ARCS, and we're going to use that video clip in our little montage for our annual banquet, or our virtual banquet, which Sarah is still working on. So yeah, I'm not totally sure what all we're doing, but we're definitely doing something in a couple of weeks.
Hey, Sarah.
Hi.
Hi.
Hi, Sarah.
Hello. Hi, Jonathan. Thanks for doing
Hi.
this today.
Thanks for inviting me.
Of course.
So I know that Kate cannot be here.
Correct.
And I'm not totally sure about a couple of people, but I think it's now 5:03. I think we can go ahead and get started.
We were going to plug in later, so okay.
All right.
Alrighty, so let me welcome everyone here. So hello everybody, my name is Patty Jones. I'm the treasurer of the ARCS Illinois chapter, and I'm also the Zoom host for our virtual events this year. So thanks everybody for being here. And as you may remember from our previous Fun at 5 talks, our protocol is that I'll introduce our speaker, and he will then take his time to give the talk. And if you have a clarifying question about something that you didn't understand, like an acronym or something short, you can put it in the text chat box, and I will interrupt Jonathan to ask him that. But then at the end of his talk is when we can all have a discussion, and we can all be unmuted and ask questions in a more free-form manner. And if you have questions that you'd like me to ask on your behalf, please put that in the chat or chat me privately, and I can ask on your behalf if you don't want to use audio yourself. So again, thank you for being here, and I'm pleased to welcome our scholar today, Jonathan Strutz from Northwestern University. As you can see on his title slide, he's a chemical and biological engineering PhD student, and as you can see, he's going to be talking about using DNA as a medium for recording biological signals. So thank you, and take it away, John.
Great. Yeah, thank you so much for inviting me. Really appreciate it. So yeah, like the title suggests, this talk is going to be about DNA and recording things. But before I get into that, I'm going to get into a little bit about who I am, and as well some background, actually a lot of background. Talk's mostly going to be background, just because it's sort of a complicated system that I'm working with. And there we go. So yeah, I'm going to start with a little bit about me. A lot of background, the problem that we're trying to solve, our solution, some progress, the progress that we've made so far, and then I'll end with some future work. So first, a bit about me. I grew up in Ohio. My interest in science began in elementary school when I mummified a chicken for a science project. I went to Ohio State, graduated with a chemical engineering degree there. I also minored in biomedical engineering. While there, I met my future wife, Ayana. She's a circus acrobat, an instructor, a dance teacher, actress, performer. I think some of you might have met her at various ARCS gatherings. And also we have two cats, Miko and Lupin. And here they are meeting for the first time, like a year ago. And then I came to Northwestern University about 5 years ago for grad school. I'm also a quadruplet. So this is me and my three brothers. So I'm this one right here. This is us now with our mom. And then finally, I'm currently a fifth-year PhD student at Northwestern in chemical and biological engineering. And I have two PhD advisors. So most PhD students just have one advisor, but I'm sort of one of the rare ones that's co-advised with two advisors. So my first advisor, he's my primary advisor, is Keith Tayo, and we do biotechnology research in his lab. And my other advisor is Linda Broadbelt. In Linda's lab, we do computer modeling of chemical reactions. And so my project sort of merges these two things, which hopefully will become clear sort of later in the talk. All right, so getting into my actual research, it involves using a special protein called TDT, which you don't have to understand what that means right now. I'm actually not even going to say what that abbreviation is because you're just going to forget it. Just know it's called TDT. And we use this special protein to record metal ion amounts into DNA. So that's probably really confusing. So that said, we need to first know a little bit about each of these things. So I'm going to talk about DNA for a little bit, then about the special protein called TDT, and then why are we recording metal ions and why are those important? So first off, as we all know, DNA basically is a set of blueprints, if you will. So it instructs your cells how to behave. So it instructs them how to live, how to eat, what nutrients to take up, how to divide, how to sense changes in their environment, even tells them how to communicate with other cells nearby. It's really complicated. And because of that, it's really long. DNA is actually made up in the humans 3 billion base pairs or letters, as you might remember them, in every single cell, and you have trillions of cells in your body, which is pretty amazing just to think about. And if you remember, there are 4 different bases that make up DNA. So DNA, first off, is a double helix, so it has 2 strands. And you might remember, if you're not a biologist from high school biology, you probably learned that DNA is made up of 4 letters or bases, A, C, G, and T. And A likes to pair up with T, and C likes to pair up with G. And so in the double helix, so at the top here we have A, which is this green guy here, pairing up with T on the right here, and then here we have a CG pair. And so DNA is really a bunch of these base pairs in a sequence, and that sequence acts as sort of like the code that tells the cell how to behave. And so one of the many things DNA provides a blueprint for in cells is creating antibodies. Antibodies, like all other proteins, are formed of building blocks called amino acids. And so the DNA basically tells the cell which amino acids to use to build a specific protein. And so you have DNA, and you might have a specific portion of the DNA called a gene that basically tell the cell, okay, for this protein, you're going to add this amino acid first, then this one, then this one, then this one, and often a sequence. And then once that chain of amino acids is built, it basically folds up and creates a special shape, and that's known as a protein. And so you can imagine if you change the DNA, that will affect which amino acids get added during this process, and that'll affect what the protein looks like and how it will function and what it does. And just for context, there are like thousands and thousands of different proteins that all do have different functions in your cells. So there are proteins that are involved in sort of like transporting things in and out of the cell. There are proteins that do chemical reactions called enzymes. And then there are proteins called antibodies, which we all know as basically as part of your immune system and protect you against foreign invaders like bacteria and viruses. And so why am I talking about antibodies? Well, basically there's sort of a problem that biology had to figure out how to solve. And that problem is there are millions of different diseases out there, different bacteria and viruses. How can we possibly have an antibody for every single one of those and store that information in every single cell? Even though DNA is made up of billions of base pairs, we don't have enough DNA to do that. And so one thing that biology did that's really smart is that cells will, when they're creating antibodies, they'll basically randomly sort of change up the DNA that codes for an antibody many times. And so they'll create lots of random antibodies. And then they'll basically have a mechanism that allows them to remember which ones sort of stick and which ones are effective against foreign hosts. And the process by which this sort of randomness occurs in the DNA is in part due to this protein called TDT. And so TDT is a protein that adds sort of DNA building blocks or letters randomly to different parts of antibody DNA sequences. So this means that after TDT adds its random bases, every antibody DNA sequence is slightly different. And this is important because that slight difference might mean that that antibody can now, be effective against a certain disease that your body is currently experiencing. But how does this actually happen? So if you imagine DNA as sort of, like we said, 2 strands. Each strand has its own letters, and you have all your different pairings here. Here I'm just showing the 2 strands as black lines just for simplicity. And so you have a top strand and a bottom strand. So DNA gets cut all the time in your cells. I'm not going to go into the reasons why, but a lot of the time it's due to basically like repair, bases that get added wrong and things like that. And when your DNA gets cut, often it won't get cut in a straight line in terms of the top and bottom strand getting cut at the same place. The top strand will often get cut at a slightly different position than the bottom strand. And so what can happen after that cut is that basically the 2 strands, so you can imagine these strands continuing on either side as well. The 2 strands will sort of float away from each other. And now you have these sort of exposed parts, which are just single-stranded DNA parts. So these parts don't have an opposite strand. And so what TDT can actually come in is, if we imagine this as part of the gene for an antibody, is it can come into these sort of overhanging parts here. And because there's no template strand here to tell what bases to add, it can just randomly add a couple bases there. And then it sort of floats away. And then through another process that I'm not going to get into, basically these strands can come back together. And now this DNA has some random bases added to it. And so that's what TDT does natively in our bodies. It's found in all mammals. So that's its function. So it adds a little bit of randomness to antibody DNA. Yeah, so now this DNA sequence has some random DNA. So we want to use this for a completely different purpose. So you can sort of forget about antibodies for now. But we know from its native function that it adds these random bases, A, C, G, and T. The last bit of info we need to understand sort of what my research involves is that TDT not only adds these bases to DNA, it needs to interact with different metal atoms to do so. So I'm depicting these metal atoms as little orange circles here. They're much smaller than TDT is. And so when I say metal atoms, I mean things like zinc, magnesium, cobalt, calcium. And so these are just little tiny atoms that are floating around in the cell, and they'll interact with lots of things, lots of different proteins and other components of the cell. But TDT specifically is one of those things that they'll interact with. And they basically help TDT carry out the reaction where it adds a new base to DNA. And so without any metals present, TDT won't do any reactions. It needs those to be present. But what's important about this is that the metal atom, the identity of it, that TDT interacts with determines how much it likes to add each base. So for example, when zinc is present, we've actually shown experimentally that TDT likes to add A's more often. Even though it's still randomly adding bases, more of those bases will end up being A versus C, G, or T. That's what this graph is showing here in the lower right. This was basically just an experiment we did in a test tube. There's just TDT floating around with some DNA and a bunch of ACs, Gs, and Ts. And we just let the reaction run. So it's adding ACs, Gs, and Ts. And we have two test tubes, one where we just have magnesium and one where we have magnesium and zinc. And so then after we ran the reaction, we sequenced out all the DNA that it made and then calculated how many, and then basically counted up how many ACs, Gs, and Ts it added in each condition. So what this graph is saying is that A was about 15% more likely to be seen in strands formed in just the magnesium condition versus C, G, and T were much more likely to get added when we added zinc. So why is this important? We can show that it does this, but why do we care about metal ions and their concentrations? So metal ions are important because, like I sort of alluded to earlier, they're involved in a lot of different biological processes in the cell. And many biological processes actually actively will change their amounts. So one of the most famous examples of this is neurons, which sort of look like this. So as you know, our brains are made up of many of these neuron cells, and they're all sort of connected together, and they talk to each other using like electrical signals, basically. And so while they're talking, people will refer to them as like firing or being on. And when they're talking to another neuron, calcium actually floods into the cell temporarily. And then as soon as they're done talking to their neighbors, the calcium goes back out. And so you can imagine if you had a way to like measure this calcium in the cell over time, you could figure out, you could basically record that into DNA and then sort of figure out when each cell was talking to its neighbors. And there's no way to do this currently. So you can like insert like electrodes to get sort of into like deeper brain tissue. But if you do that, one, it's really invasive. Two, it's not high throughput. So you can only do this for a couple of neurons at a time. But if you had a system like this where you could just make it so every cell naturally, produced a bunch of TDT and DNA to add to, you could potentially use that as a system to record sort of the state of each neuron over time. And if you could do that, you could basically map how the neurons are connected together, which we currently don't have sort of resolution at the single neuron level. And this is just one application of this TDT system. There's many others where metal ions are present, or metal ions are important. I just wanted to highlight this one because I think it's maybe one of the easier ones to understand. And so this is sort of an example of what an experiment might look like. So you have some signal. So this could be a neuron being on or off, which is equivalent to the calcium being really high or low in the neuron. And then so maybe in this neuron, we have this TDT system, and over time it's creating DNA, but the DNA that it creates looks a little bit different over time based on whether the calcium is really high or not. And so for the first part of the DNA, when there's very low calcium, you might have, say, like not very much A's and C's. But then for the next portion where calcium is really high and the neuron's talking to its neighbors, you might have a lot more A's and C's. And so you can sort of divide the DNA into different chunks based on sort of the ratio of A, C, G, and T in the DNA. And so what we're really doing is using TDT as a sort of a recording device to track different metal ions over time. And so now I'm just going to talk a little bit about what we've actually done so far. So that's sort of the dream. We've run a lot of reactions in test tubes without any cells. And we do this because it allows us to isolate TDT by itself, and we can study it without all the other interacting effects of the cell, which is really complicated, has thousands of different moving parts. And this, just helps us understand TDT and how it works. We have also run some initial trials in cells. We're currently trying to measure changes in intracellular zinc, just because that is sort of giving us the biggest change in the ratio of ACG and T currently. So for neurons, we would want to measure intracellular calcium changes, but so far we haven't seen a huge shift in ACG and T sort of incorporation rates compared to things like zinc and cobalt. So that's why we're choosing zinc first. We've had some modest success so far, but we're still working on that. And there's a lot of challenges still ahead. So even if we do see a signal in the test tube, and even in early experiments with cells, once you get into actual neurons, it could be completely different because there's many different cell types in the body. We're currently doing these sort of early trials in I forget what kind of cells. I think they're breast cancer cells. And we use those because they grow really fast. But obviously neurons are really different, and so there could be a lot of differences with that if we want to use this specifically for neurons. The signal strength might change, for example. So maybe in neurons, there's other interacting factors that make it so we just can't see any change in the ratio of bases that gets added. There's other systems in cells that could interfere with our recordings. There are special enzymes in cells that like to just go around and chop up DNA, especially if it doesn't have a template strand. So if it's just a single strand and doesn't have a partner, they'll just like chop it up. There's also a lot that's unknown about how TDT actually adds bases randomly, what influences, like in some conditions it'll have a 25% chance to add an A, but maybe in other conditions it'll have a 40% chance to add an A. What are those conditions and how much effect they each have? So I'm working on developing a computer model of this reaction in a test tube to basically better understand how it works and answer some of these questions. And so in terms of future work, in the near future, we're going to continue with trials in cells. I'm going to work on specifically building out the computer model of this reaction and try and answer some relevant questions. So if you have questions about what those questions are, feel free to ask. And also, our lab is currently working on changing TDT itself. So you can imagine, so TDT is a protein itself, and so it has its own DNA sequence that encodes it. And so if you change that DNA sequence, that can slightly change how TDT functions. Most of the changes that you make to the DNA sequence will just break TDT and it won't do anything. But there are some that you can imagine might improve or make it add way more A's in a specific condition versus another, way more G's in a specific condition than another. We're also looking at getting TDTs from different organisms. So across different organisms, so for example, in mammals, which all have TDT, it's not the exact same version of TDT. There's slight differences in the DNA sequence, even though they all do roughly the same thing. And so all the work so far has just been with cow TDT, because that's the one everyone uses for some reason, and human TDT as well. But you can imagine that, because there's so many different organisms out there, of course, we should look at other TDTs and other organisms and try and see if those have better sort of properties for recording. And so Maria in our lab is working on this, and she actually even had a TDT from axolotls, which are like the cute little amphibians in Mexico that are like going extinct, unfortunately. And it's a cold-blooded organism, so actually maybe its TDT is like completely sort of different, maybe has different behavior at different temperatures than cow or human TDT. And then in the far future, hopefully we could end up with a highly non-invasive system for recording metal ions over time, such as calcium and neurons. So that's all I had. I guess I'd just like to acknowledge my PIs, Linda and Keith, the TDT team. So Namita, Maria, and Alec are all the experimental people on this project. I'm the sole computational person, so I like basically analyze all the DNA sequencing data, build models to try and answer questions, things like that. The Broad Belt Lab, which is pictured on the bottom here, and then my wife and our 2 cats. And of course, ARCS as well, and my other funding sources. So with that, I'll take any questions.
All right, virtual applause. Thank you so much, John. One question I'll ask you in general before we get to more technical questions is, could you tell us more about how ARCS helped you in like, what did you use the money for, and how was this support helpful for your research?
Yeah, one of the main things I used it for was actually not for this project, but for my other project, which I gave a talk on like 2 years ago. It allowed me to sort of travel to a conference and meet a bunch of people. And actually one of those people I met, I ended up on a publication with as a second author. And it probably wouldn't have happened if I hadn't gotten the ARCS money. So that's one thing. In terms of this project, I haven't used any of the ARCS money on this project specifically. But yeah, on my other projects, it's been a real help, especially for collaborating with people I might not have collaborated with otherwise.
Yeah, that's great to hear. I mean, I think that's our intention, is that, you're usually, the students are funded by grants, and so you have a very specific plan of work and things you're supposed to be doing, but ARCS is meant to give you that boost and go beyond that. So thank you for that. That's great. That's great. One question I see that from the chat to me was questions about how metal gets into cells, and even more specifically, this is maybe getting into nutritional science question of, if we eat a lot of a certain kind of metal, how does it get into our cells, and does that affect this process at all? Or is this maybe a bit too beyond what chemical engineering does versus
Probably. I do know. So cells are basically these blobs, and they have an outside called the membrane. And in that membrane, there are proteins which act as little pores, so like cylindrical proteins. And a lot of those will basically act as transporters and allow metals in and out. And even some of them will basically say, okay, I'll take 2 sodiums in and I'll release a potassium out every time I do that. And so there's lots of different like transporter proteins that transport metals in and out. As far as the other part of that question, I'm not really entirely sure like exactly what the effects would be if you ate like a bunch of a specific metal. I imagine it would just get excreted, just because your cells can only take up so much, and they have mechanisms for sensing how much is in your cells. And at that point, they'll just stop bringing things in.
Okay. I had another question about tracking, metals in cells like in neurons. Because so I work at Beckman Institute here at U of I, which has MRIs and a lot of neuroscientists. So there's been a number of efforts related to imaging of live cells, in vivo imaging. And so I was wondering, what's the sort of the use case for why you would use this approach versus, say, have a live animal or human go into an MRI scanner and look for iron. I guess you can't look for everything with an MRI scanner, though.
So correct me if I'm wrong, but my understanding is that MRIs can't get you like single cell resolution, whereas this system could, because you have a separate sort of TDT and DNA recording in every single cell.
Yeah, I think that's generally true. I know that some of the electrical engineering professors here are working on amazingly high resolution scans, but I don't think they're certainly sub-millimeter, but I don't think they're cell level particle, like nanoscale or whatever. But yeah, that's an interesting point that, if we are able to bring all these things together, then we would have a really good picture of, the entire brain working.
Yeah, it's an important problem.
Which is the dream of many people. Yes. Oh.
Yeah, there's a lot of different approaches that people are trying to take, which is a good thing.
Okay, let me open the floor to other questions. First, let's have some maybe technical questions about the talk, and then I'm sure that other people have questions about growing up as a quadruplet and that kind of thing too. Well, let's start off with some of the questions about the talk itself. Go ahead, Sarah. You can turn.
Yeah, I guess I have to be the one to ask. What are those questions that you're gonna work on in the future work?
Oh, I'm glad you asked. I actually have a backup slide for that.
Not planned.
Not planned at all. So if you imagine this as sort of your DNA strand, and each color represents a different base, and it's single-stranded because TDT adds to single-stranded DNA, so there's no partner here. You can imagine that maybe the previous bases it added, like at the end here, could affect the identity of the next base, which we've actually have some evidence that that's true. And so we're trying to figure out how that happens. So the enzyme binds at the end of the sequence. Does it like to bind to specific sort of subsequences here? Or does it just bind to all sequences sort of generally, but then depending on what's there, it'll add different bases at different ratios? Another question that I'm looking at is, because single-stranded DNA has all these bases here, and these bases aren't connected to anything like they would normally be on the other side, it can sort of fold up on itself and connect to itself. And so you can imagine there could be specific sequences where that makes this final part of the sequence inaccessible to TDT. So if this part like folds up back over here, then TDT might be able to not be able to access it. And so I'm also using the model to determine, like, is that a major effect that we see, and how much could that affect how much sequences get extended. And then one of the other questions I'm looking at is, does TDT stay on the DNA strand after it adds a base? So it could add a base, then stay on the strand, then like sort of move a little bit on the strand to the next one, add another base. And if it does that, it could do that really fast. And that's actually how normal, they're called DNA polymerases. So the enzymes that create DNA normally to copy it, so that when your cells divide, each cell has a copy of it. That's how they operate. They basically will attach to one of the two strands, and then or attach to both of them actually, because it has to know the template strand, and they'll sort of run along it and just add bases really, really fast. But TDT is thought to sort of just fall off after it adds every base. But we actually have evidence that shows maybe it doesn't do that after every base. Maybe it does it after every few bases. And so yeah, those are some of the questions that I'm looking at, which no one's really answered up to this point. And I'm using the model to try and investigate those questions. So basically, you can use the model to say, if this is true, based on our model, what would we expect to see? And then we can compare that with the data that we have.
Oh, thank you. I do have a question about your model. Is this like a biophysics model that's like atomic scale, or what?
It is
what
a
model?
kinetic model.
Okay.
So it's focused at the reaction level. So this probably isn't going to make sense to most of you. But basically, it has sort of math equations that describe all of the possible reactions. So it could be adding an A, adding a C, adding a G, or adding a T. But then you can even expand that to like adding an A to an A, adding an A to something ending in a C, and so on. And you can go even further back, adding an A to something ending in AG, and all of those combinations. And so it's looking at that primarily, yeah. So it's sort of like the chemical engineering side of it, where people use like kinetics, reaction equations basically to describe systems like this.
Yeah, I mean, there's always this modeling question of like, how low do you go and how much computational effort you want to put in to get a result. So I know that the group at Beckman that does computational biophysics, they will spend like 150 million hours on a supercomputer to simulate a few nanoseconds of some reaction because
yeah, this is not that detailed
every
at
single
all.
element, its vibrational state and all this other stuff. So yeah, if you gotta be, so yeah, you can model things at the higher levels and get much faster results.
Yep.
All right, let's open the floor to more questions. Does anybody else, go ahead, Sarah, you're waving. Go ahead.
Sandra actually had her hand raised. I'll let
Oh,
her.
sorry. I don't see you on here. Okay. Go ahead, Sandra. Oops, where is she?
You're muted, Sandra.
Oh, I've just. Oh, there
I
you
didn't
are.
have a question. I was moving my screen.
Oh, okay.
Okay, well then I'm gonna ask another question.
Okay.
How has COVID affected your research?
As you can imagine, I've been working from home, which is possible because I'm computational only. So our lab is roughly half experimental, half computational. So all the experimentalists have gone back at this point. They actually alternate each week, like who's in the lab. But for all the computational people in the lab, we've just been working from home. So that's made it a little bit more difficult, I would say, just to stay motivated, because I find that I get more motivated when I'm around other people that are working on similar things, which is probably a common thing. Yeah, I think that's the major change. In terms of like funding or stuff like that, hasn't affected anything. Yeah. Good question.
So I do have a question. When are you going to graduate? Do you have a timeline for that?
Yeah, my timeline, it said winter of next year, but I think it's probably going to be spring of the year after that. Just because, I don't know, things always get pushed back for one reason or another.
Right. Are you looking at academic jobs or industry jobs or what?
Industry jobs. So I actually did an internship with a company called Lonzatech in Skokie.
Oh, okay.
They don't do this sort of stuff. They actually do something completely different, where they have bacteria that eats waste gases like carbon dioxide and carbon monoxide and produces useful things like ethanol and acetone, useful things in the chemical industry.
So that's what you're thinking, is that you're going to do like a environmental biofuels sort of a thing?
Yeah, so they have a computational biology team, which is the team I worked in.
Mhm.
And so during my internship, I did some modeling for them to try and predict like, if you make this change to your organism's DNA, it'll be able to produce, more acetone or more ethanol, stuff like that. Yeah, so I like doing that stuff. I also like this DNA recording project, but I'm probably not gonna do much in this space after I graduate.
The DNA stuff seems much more university sort of a thing.
Yeah, there's a handful of companies working on this, but it's much more sort of in the academic space.
Right. Any other questions from the audience?
I'll ask one more. Sorry.
Okay, go ahead.
Jonathan. So your previous research was similar to what you did your internship with, correct?
Yeah.
And then how
Yes.
did you shift to this work?
Yeah, actually, so yeah, I have two projects. So this is one of them. The other one is the one I talked about like 2 years ago, which is more similar to what I did during my internship. And so I've actually been working on them sort of in parallel. I haven't really been jumping, or it's not like I completed one and now I'm doing the other. So that makes it sort of difficult, but it also means I always have things to work on. And the reason I originally was able to work on this project was because I was accepted as a BTP trainee. So BTP is the Biotechnology Training Program at Northwestern. And basically it's a training program funded through the NIH. And it's at a bunch of different schools, including Northwestern. And they'll basically take a handful of people every year to give them funding for a year or two. As a requirement, you have to do an internship, which is actually why I did an internship. And it has to be an industry. And it's basically, its mission is to train people to go into like the biotech industry. And so as part of that, because my funding was no longer centered specifically around my original project, which I talked about 2 years ago, I was able to branch out a bit. And I told my advisor I was interested in this project, especially because the person who was doing all the computational work for this project was graduating. And so I sort of took his spot, I guess, after he graduated and filled in for him, and then got more involved after that as well.
That's great. Thank you. Okay, so let me ask you about the mummified chicken and what it was like to grow up with as a quadruplet.
Yeah. So 5th grade, mummified a chicken for my first science fair project. And I did that because I was obsessed with Egypt for whatever reason. Yeah, I remember basically, I didn't actually mummify like any chicken. It was just like a chicken from the grocery store, but I basically preserved it using mummification. And so I didn't put it in a fridge or anything. I just sort of left it in our garage and mummified it. And every, day or two, I would have to like, change different things about it. I have to add more salt and stuff like that. So yeah, it was an interesting project.
And were you and all of your brothers also interested in science, or were you all pretty?
That's a good question. So me and Tim are both engineers. He's a software engineer though, and he works for a company called Booz Hamilton, and they basically do contract work, a lot of contract work with like the U.S. military and other companies. And then Andrew and Chris are both accountants. So 2 and 2, I guess.
Interesting. Oh, that's great. All right. So does anybody else have any other questions? Sarah, did you get what you needed from John about videos, what he said earlier about what ARCS is? All right. So yes, we have our content ready for our other reel that we're going to do later on. Okay. Anybody else have any questions? I see a couple people have joined in later. So I'll hold the floor open for another minute or two here. Okay, well, thank you again so much for that wonderful presentation, John. Again, applause. And I think this is wrapping up our series for the year as well. So again, thank you so much. We are very proud and to have supported you, and we're really pleased with the excellent work that you're doing. So keep it up, and we will be, of course, you'll never be rid of us. We're going to keep track of you as an alum throughout the years. So,
Awesome.
so we will stay in touch, and please stay in touch with us as well.
All right. Thank you, John.
All right. Thank you very much. We'll end the meeting now. We are adjourning. Thank you so much, everybody. Take care. Bye-bye.
Bye. Thank you.
Take care.